Bismillahirrahmanirrahim. Assalamualaikum everyone. Welcome to Stream of Knowledge. Today we will discuss about shape and size of bacteria. This is our first lecture on basic bacteriology. Bacteria are classified by shape into three basic groups: cocci, bacilli, and spirochetes. Cocci are round, bacilli are rods, and spirochetes are the spiral-shaped bacteria. Now the question is, why bacteria have specific shapes? And the answer is that they contain rigid cell wall. Actually, their cell wall determine their proper shape. But there are also some bacteria that do not contain the specific shape, means they are pleomorphic or variable in shape. For example, mycoplasma. Mycoplasma does not contain cell wall, so it does not have any specific shape, so it varies in its shape. Now the question is that why it is important to study the bacterial shape? The answer is that for the proper identification of bacteria, it's important to study about its shape so that we can come to know about its morphology and microscopic identification. Moreover, the shapes arrangement of bacteria is also important for its proper identification. Now we are going to arrangement of bacteria. Okay, arrangement of cocci. Cocci, if it is a single, then it will call coccus. If it is in pair, then it will call diplococci. Di mean two. But if it is arranged in pairs in single plane, as you can see that if it is arranged in single plane, remember the plane of division is very important while discussing the arrangement of cocci. If it is in single plane, then it is called as diplococci. For example, Neisseria species and strep pneumonia species. And if it is the pair but in two planes, then it will be called tetrad. But if it is the pair of bacteria, pair of cocci in three planes of division, then it is called sarcina. But if it arranged in multiple planes and having like a group or a grape of like clusters, then it is called staphylococci. For example, staph aureus. And if it is arranged in chains, then it will be called streptococci in single plane of division. For example, strep pyogenes. Now moving toward the arrangement of bacilli. If it is single, then it is called bacillus, and if it is arranged in single plane of division in pair, then it is called diplococci, as di mean two. Strap mean chains. One thing you should remember: staph mean cluster of grape-like cluster, and strap mean chains. Here you can see it just like a chain. If it form a chain, then it is called streptobacilli. And what one thing you should remember: there's difference between streptobacilli and palisades. Although both of these are chains. Palisades are also chains and streptobacilli are also chains, but the major difference is that they are connected by end to end while they are connected by side to side. You can see that bacteria are connected side to side, and if the chains are present in that, like bacteria are arranged in side to side, then it is called palisades. Cocobacilli are also bacilli, but they are short in structure, and moreover they look like a cocci. For example, Yersinia pestis. And now we are going to discuss about the arrangement of spiral. First, very common, spirochetes. Spirochetes are have coils, but these are tightly coiled, while spiralia have loose or relaxing coils. Spirochetes examples are Troponema palladium, Borrelia burgdorferi, Leptospirilla, etc. While spirilla or these are helical shaped, and these have one thing you should remember, that spirochetes are the tightly coiled while spiralia are the loosely coiled. For example, Troponema palladium have tight coils while Borrelia burgdorferi have loose coils. And one is Vibrio. Vibrio has only single curve, or another example of single curve bacteria is Helicobacter. This was all about bacterial shape and arrangement. Now we are going to discuss and apply our knowledge. Okay. Before identifying the bacteria, you should remember there are three basic shapes: cocci, bacilli, and spirochetes. If they are round, then it is cocci. So in the first row you can see all are round, so all of these are cocci. And if they are arranged in grape like clusters, so that will be the staph. And if they are in like chains, that will be the strep. And if they are, these two both are diplococci as they are arranged in pair. Remember, there are two diplococci. One is Neisseria and second one is strep pneumonia. What is difference between two? If they have pointy dents or oval shaped, very important terminologies. If they are pointy, if they have pointy dents or they are oval shaped, then it will be the strep pneumonia. But if it arranged in kidney shaped or kidney like beans, then this will be the Neisseria. Okay, now we are going to discuss about bacillus. This complete row are the bacillus as they are rods. This will square shape, so this is bacillus. Bacillus are the square shape, and these have round ends, so these are salmonella. This is club shell corynebacterium, and these are fusobacterium. Sorry, this is not including bacillus species. These are vibro as they are in coma shaped. And these are what these are relaxing coils, so these are spirillum, and these are tight coiled, so these are spirochetes. Now moving towards the shape of bacteria, size of bacteria. Bacteria range in size from 0.2 to 5 micrometer. The smallest bacteria or mycoplasma, which does not have its cell wall, are about the same size as the largest virus or pox virus. But the longest bacteria rods are the size of some yeast and human RBCs, for example, 7 micrometer. But the bacteria range in size from about 0.2 to 5 micrometer. That was all about the size of bacteria. Thank you for watching. Do subscribe, like, comment and share, and stay tuned for the next series of basic bacteriology. In next session, we will discuss about the structure of bacteria regarding peptidoglycan, lipopolysaccharide and flagella, plasmids, etc. And all the series of we will complete all the series of basic bacteriology. Thank you.